Our next presentation is going to be ReNeuron. Michael Hunt is going to present for the company. And again, we're going to be talking about stroke, but we're also going to be talking about an allogeneic model and, another really innovative cell type. So let me invite Michael. Hi, how are you? Nice to see you. And by the way, this is a company, you really pulled a stem cell rabbit out of your hat this year. You raised how much money over abroad?
Well, last year we raised equity and non-dilutive money just shy of 60 million US.
Yeah, so very significant. It's interesting to pay attention to this company. Thank you.
Okay. Thanks very much, and thanks to the organizers for allowing me to present today. One of the few non-US companies on the roster, so without further ado. So we are a UK-based clinical stage cell therapy business. The business was founded around technology we've been working with for over a decade now, which is a highly scalable way of expanding stem cells allogeneically. So this is another off-the-shelf allogeneic approach we're targeting here. We're targeting three principal therapeutic areas at the moment: stroke disability, CLI, and retinitis pigmentosa. We've attracted a pretty strong generalist and specialist life science institutional shareholder base. Invesco, Abingworth, and the Welsh Life Science Investment Fund now constitute the three major investors on the register. We are a public company quoted on AIM in London, and as mentioned, we now have a solid balance sheet with the business funded right out to Q4 2016. The image is a snapshot of our lead cell therapy product, CTX. And that forms one of three technology platforms we're looking to exploit. CTX is the most advanced platform. It's a neural progenitor cell line. It was derived over a decade ago using an immortalization technology that we've been using since that time. It's patented and gives us the basis of a low cost of goods, highly scalable allogeneic cell therapy candidate, cryopreserved right the way through from master cell bank through to drug product stage. It offers a degree of guaranteed product consistency because it is a continuous cell line, so we have no need to rederive, so there's no batch-to-batch or patient-to-patient variation here. And we've scaled that under GMP conditions with outsourced contract manufacturers. The second platform is a retinal progenitor line, HRPCs. This is a non-engineered line, and it's expanded in a low-oxygen culture technology that we've licensed from the Ischaemia Eye Research Institute at Harvard. Again, it's an allogeneic approach to cell therapy, and again, we're in the process of scaling that to GMP at the moment with an outsourced manufacturer here on the East Coast. Our exosome technology or platform is more recent, so it's less than a year old, and this really is harvesting some of the byproducts of CTX cell manufacture. These nanoparticles, known as exosomes, which we believe have therapeutic potential in their own right if we can purify them, as we're in the process of doing. And we hope to be able to take these particles into a number of new indications beyond those that CTX itself, the cells, are being targeted at. And we have some early positive preclinical data to support that. We put a lot of patents around this technology over the last year as well. And the good news here is that CTX, as a continuous cell line, is a very potent producer cell line for these exosomes. And I'll come back to that platform briefly towards the end. The pipeline looks like this. So dark blue tells you where we are with these programs. Light blue shows you where we hope to take the programs over the next 2 to 3 years with the money that we now have. So we're pretty well funded to take the two core CTX-based programs through to clinical proof of concept, and the retinitis pigmentosa program and the exosome program following on behind there. So moving on to the therapeutic programs in turn. So REN-001 is our program targeting stroke disability. So it's important to note that this is not an acute stroke treatment to keep the patient alive. This is addressing the consequences of stroke several weeks down the line. As you may have already heard today, stroke is the single largest cause of adult disability in the developed world. It's a very large unmet medical need, around 800,000 stroke events per annum here in the US. And health and social care cost bill that goes with that, very significant. No pharmaceutical treatment options beyond outer plays, which has to be administered very, very shortly after the infarct. We've independently analyzed and tried to stratify the patient population that we believe would be the best responders for this intervention, and on reasonably conservative assumptions, that offers a market opportunity for us, we believe, of somewhere between 1 and 2 billion per annum, again here in the US for REM1, in that targeted sub-patient population. We and our collaborators have published extensively preclinically, both in terms of the efficacy of CTX cells in validated stroke models and the mechanism of action that we believe are at work here. And again, in common with other cell types, this is a multimodal trophic cell therapy approach. Specifically, angiogenesis, neurogenesis, and immune modulation are the key mechanisms we believe are at work here in the brain with CTX cells. But the important thing here is that REM1 looks to target to improve the trajectory of recovery in disabled stroke patients around 8 to 12 weeks post-stroke. That, we believe, is the sweet spot for administration of this treatment in these survivors. So we're just through a phase 1 trial now that we ran in the UK. It was a single-site dose escalation study in 11 patients on a 2-year follow-up. The procedure itself is a minor neurological or neurosurgical procedure that takes around an hour to 90 minutes, and the patient typically stays in overnight or perhaps 2 nights. And what you see here are a number of fairly classic stroke measures used in efficacy studies, and we deployed these really as to monitor patients for lack of deterioration for safety purposes, and also to evaluate them for potential efficacy or use in efficacy studies thereafter. And we were pleased to see actually a fairly sustained improvement in pretty much all of the patients on most, if not all, of these measures over time against a baseline steady state deficit. So these patients were monitored for a steady state deficit for 10 weeks prior to transplantation. And just as an indication, the Rankin score is a measure of disability and handicap. A 1-point drop on that 6-point scale would be indicative of meaningful clinical benefit to the patient. Something, for instance, that might take the patient from assisted living to assisted independent living. So something that's really meaningful in terms of the patient and perhaps for a healthcare payer in terms of reducing the reliance of these patients on health and social care. So an encouraging start for the program, and leads us into phase 2, and we now have regulatory approval for a phase 2 study in the UK. It's been adopted by the NHS Stroke Research Network. So in the UK, the NHS is very well connected, and those UK sites are incentivized in terms of promoting innovative clinical trial activity at those hospitals. They're incentivized to recruit at an appropriate pace and so on. So we're leveraging into that infrastructure that exists and that connectivity. We're going to start this trial in Q2 very shortly. It's an adaptive trial design in up to 41 patients across 10 sites, really to confirm a meaningful efficacy signal ahead of a larger pivotal efficacy study thereafter. And the patients really will need to suffer a paretic arm 4 to 8 weeks post-infarct. So it's a unilateral deficit which manifests in motor deficit, motor disability on one side of the body. And if you look at the time course of recovery of stroke patients, if they survive, their chances of any further spontaneous recovery beyond that 4 to 8 weeks are very small indeed. So we're looking, as I said previously, to improve that trajectory of recovery using REM1. So endpoints will be using the same as we used in the phase 1, but with the addition of the ARAT test, the Action Research Arm test, which is a very clear clinical measure of the patient's ability to perform a useful task with the affected side of the body, with that affected paretic arm post-treatment. It's been part funded by a UK government grant, and alongside we're running a non-interventional observational study at the same centres using the same inclusion criteria to do two things. Firstly, to pre-screen patients for the interventional study, and secondly, to build a valuable clinical data set in the patient target population that we're looking to treat with REM1 per se, and those data really don't exist at the moment anywhere in the world. So much for stroke. Moving on to CLI, which uses that same CTX cell line. Critical limb ischemia, as you've probably heard already today, another very significant unmet medical need, a loss of blood flow to the lower limbs, leading ultimately to dry gangrene, which is what you see in the image here, and consequent amputation. Common in diabetics, and again, a very clear unmet medical need with the costs that go with that. No current treatment options except surgical revascularization, for which only a proportion of patients will be eligible. So again, independently analysed, we estimate a market value for REN9, if successful clinically, at somewhere between, coincidentally, $1 and $2 billion in the US. And again, we've published, in conjunction with our UK collaborators at the Bristol Heart Institute, efficacy data showing the very clear and dose-dependent pro-angiogenic effects of CTX cells in lower limb, hind limb ischemia models, this restoration of blood flow. And again, the target here quite simply is to improve blood flow, leading to a reduction in incidence of death and amputation in these patients. A very straightforward clinical development strategy here. We now have regulatory approvals for a phase 1 study, which we're going to start next month. In the UK, again, it's been funded by a UK government grant or part funded by a UK grant. in up to 9 patients at a single center, another dose escalation study, which we hope will be followed in the first half of next year by a classic phase 2 double-blind placebo-controlled study, probably in around 80 patients, looking at both US and European sites, with a composite endpoint of reduction in death, amputation, and also doubling rates in these CLI patients. The final cell-based program is our program using the HRPC cells for retinitis pigmentosa. And again, we have a patent on a production process developed by Mike Young's lab at Scapens that permits large-scale cell bank generation of these cells. And again, we continue to work with the guys at Scapens, as well as more recently now the Institute of Ophthalmology at Moorfields in London. The first target is retinitis pigmentosa, which is an inherited blindness-causing disease. The image here is the world as an RP patient would see it, this gradual loss of peripheral vision leading to tunnel vision. We have orphan drug designation for this treatment, both in Europe and in the US. Target indication value over here is somewhere in the order of 200 to 400 million per annum. And we're currently completing IND-enabling studies with a view to starting a phase 1/2 by the end of this year. A little bit of preclinical data on the retinal program. Again, using the classic RCS, Royal College of Surgeons dystrophic rat, we see a very clear evidence actually of the HRPC's ability to prevent loss of visual acuity in these animals. We've also generated a homologue, a pig retinal progenitor cell, which we've engrafted successfully into pig eyes, and we see very good engraftment and connectivity through to the photoreceptor layer. And differentiation as well into photoreceptors. So we are looking for a degree of both trophic effect here, and also we hope differentiation into those lost photoreceptors prevalent in RP patients. Finally, a quick word on the exosome platform. Again, this is leveraging the power of CTX, if you like, and harvesting from the conditioned media these nanoparticles and purifying them. We're looking at preclinical targeting in a number of new areas where we're not taking CTX, including wound healing, immune modulation. We see neuroprotective effects of the exosomes and also suppression of cancer cell proliferation as well, which we're very excited about. And cancer, more than one cancer indication, will be one of the prime clinical targets, we hope, with this new platform in due course. We put a lot of patents around this, composition of matter, characterization, manufacture, and therapeutic use patents in this technology. We've also attracted some Welsh Government money to support the preclinical development. And that leads me on to funding. And as mentioned at the start, we had a pretty good year last year, considering our prior history up to that point of raising money. And it was done in quite a strategic way. Initially, we set out to do what you see in the middle part of this slide, which was to garner some fairly significant grant money or non-dilutive money from the Welsh Government to build out our manufacturing capability, first and foremost, for phase 3 needs and beyond. And that facility we're about to sign a lease on the building in South Wales, and it will be ready for GMP manufacture from 2016 onwards. Having got that, we then went on and garnered a number of TSB, Technology Strategy Board UK government grants, just under 5 million worth, match-funded grants that involve fairly heavy due diligence. And having garnered this soft money, we then went back to our equity investors to raise the equity money just over 40 million. So that made up the package of just shy of 60 million US equivalent. And as I said, that's really given us the ability to take our programs through to what we hope will be meaningful value inflection points. And I've listed those out here. So as you can see on the CTX-based programs, a fairly sort of continual flow of clinical data out to clinical proof of concept in phase 2 over the next 2.5 years or so. The retinal program, again, into its phase 1/2 stage, and all being well, the exosome program showing its therapeutic potential clinically within this period as well, at least at phase 1 stage. And I think I'll leave it there. Thanks very much for your attention. And I'll be up on the 5th floor if anyone wants to meet up.